everyone. My name is Divya, and I'm representing MIT iGEM. And today I'd like to tell you about a novel technique called RNA strand displacement that can be used as an information processing mechanism in mammalian cells. So at the core of synthetic biology, we're really looking to engineer circuits we're looking to engineer sophisticated circuits that can help control cellular behavior. And traditional approaches have relied on improvements in the fields of DNA synthesis and assembly, which combine multiple transcriptional parts in order to encode for higher logic functions. But we see a limitation in this technique, in that our transcriptional units are incredibly large and require significant amounts of DNA to encode them. Our approach offers a complementary strategy, which allows us to use smaller parts. Given an amount of DNA, we can encode for a higher number of logic functions, and thus we can enable greater sophistication per unit of DNA. And this property is incredibly helpful. However, in certain cases, it can be absolutely critical. We can improve our delivery of our circuits to cells in cases where payload of DNA is limited, such as using viral delivery. And furthermore, we can lower the energetic penalty that our cells suffer when operating the circuitry. So in the past decade, while we've seen recent improvements in terms of growth of these circuits, we've ultimately seen that the growth has been slow when using transcriptional-based topologies. In contrast, we've seen exciting developments in the field of DNA computing and in vitro strand displacement. And we've seen remarkable growth in a very short period of time. However, these technologies have been limited to an in vitro context. And for the remainder of this talk, we'll be referring to in vitro as computation that's carried out in solution or outside of a cell, whereas in vivo is computation that we can engineer within cells. So our goal for the summer was to demonstrate that strand displacement is a viable technology that can perform computation within cells, or in vivo. But to implement more sophisticated circuits, we first need to build simple logic functions. Today we're going to be showing you the implementation of a simple buffer gate in which an input signal is processed into a matching output. We will then move on to show you a novel construction of a NOT gate, in which an input signal is inverted. And our technology supports the development of higher logic functions, including OR and AND functions. So to implement a simple buffer gate, we can see an analogy in terms of traditional transcriptional circuits by using an inducible promoter that drives subsequent gene expression. In the case where we have a low concentration of our activator, our input signal is low, and thus our inducible promoter does not yield an output signal. However, in the case where we do have a high activator, our input signal is present and allows the inducible promoter to express our protein and yield a high positive output. And we can see how this analogy is translated in the context of strand displacement, where we will denote a high signal to be a free strand of DNA that is able to interact with other strands, and a low signal as a double-stranded DNA complex, in which our green strand of interest is bound to a black gate complex. Furthermore, we can use fluorescence as a means of detecting cellular levels of bound and unbound signals. For example, we can conjugate a fluorescent dye onto our strand of interest, such that when the strand is free, we see fluorescence. And we can conjugate a quencher onto our gate strand, such as when the strand is bound, our fluorescence is quenched, so we do not see a signal. So in the simplest case of the buffer gate, when our input signal is 0, our output signal is also 0. And this is because our strand of interest is complexed with our gate. However, in the case where our input signal is present, we will show that strand displacement provides a viable mechanism by which this input signal can titrate away the gate to yield a positive output signal. So let's investigate this mechanism more closely. We first start with our gate-output complex, which is bound together by this hybridization domain. Furthermore, our output signal has an overhang sequence. We also have an input strand, which has a similar complementary hybridization domain, and it has its own overhang sequence. And to give you an idea of how small these parts are, we see that our hybridization domain is simply 15 nucleotides long, which is significantly smaller than normal transcriptional units. So let's see what happens when these two strands try to interact. We see that nothing happens, and our input strand is unable to displace our output strand. And this is a result of the fact that the gate-output complex is very thermodynamically stable.
So we need to get these guys to interact. One way we can do this is from a paper published last year, where you add a small unbound region called a toehold, shown here in yellow. This region changes everything. It allows the input signal to bind on the yellow region and displace the output signal, shown here. So now our output signal is free. Now what's really cool about this is the output is in the exact same form as the input. So it is allowed to be a separate input signal for another reaction farther downstream. Here, we cascade 2 buffer gates to show you how it would work. The output from the last reaction comes in, binds in the same manner, and displaces the next output. This was just 2 buffers, but you can imagine we can cascade any logic gates and scale up to incredibly sophisticated circuits very easily. So now that we have this great mechanism, we need to put it inside the cells and inside synthetic biology. What this means is that we need to sense inputs, process information, and actuate responses, all while producing these circuits inside the cell. Let's talk about processing information first. We already have an AND and OR gate, which allow us to compare inputs from the same paper. The AND gate produces 1 if both of its inputs are 1. The OR gate produces 1 if any of its inputs are 1. What we don't have is a NOT gate. These 3 gates together complete a fully complete logic set, which allows us to produce any calculation. There was no NOT gate in the literature that was compatible with Strand displacement, so we had to design our own. This is what it looks like. It has 5 parts. It has an input strand, a dynamic gate, which we've called A, the output, which we call B, buffer C, and the fuel and catalyst, which is D. The most important are the first 3. We're going to talk about these. But if you want to really know how it works, you can read about it on our wiki. So again, the output takes a low input signal and maps it to a high output signal, or a high input signal and maps it to a low output signal. In the first case, we have low input, and our output is free to react with the dynamic gate. This reaction is reversible. So if there were another reaction farther downstream, the output would be free to bind with that. In the other case, you have high input, and you want to map it to low output. Here we have an input strand, which binds with the bottom part of the dynamic gate. The output strand binds with the top part of the dynamic gate. This reaction is irreversible, so our output is trapped and can't react farther downstream. Creating this was no easy task. First, we came up with several designs and simulated them in Visual DSD. Here we have output on the y-axis and input levels on the x-axis. The ideal NOT gate would have very high output at low input and very low output at high input. As you can see, our purple design didn't work too well, so we scrapped that and stuck with the red design. We ordered it and ran a plate reader test. This was our first result. It's not great. We went back to the drawing board, and from our simulations, we knew that by varying the concentration of A, the dynamic gate, we could get more NOT gate-like behavior. So after some more tweaking, we changed the concentration both relatively and absolutely. We got this. This result shows our in vitro NOT gate transfer function, which not only matches our simulation very well, but behaves as a very good NOT gate. So this is great.
Now that we have a fully functional set of logic gates, we need to think about how we want to use these circuits. All previous work in the field of strand displacement circuits has taken place in vitro, or outside of the cell, but we want to be able to put it in vivo. To do this, we utilize RNA instead of the usual DNA, because RNA can be produced continuously, and it enables dynamic circuit function. In addition, we chose mammalian cells because they have a slower cell cycle, which means that any parts we put into the cell are diluted at a slower rate than if we put them into a bacterial cell. We can also interface with the natural RNA interference pathway, and RNA is more stable in mammalian cells than in bacterial cells. All of our in vivo experiments took place in HEC293 cells. After mining the literature, we found that the U610 promoter is a good candidate for producing short RNAs in mammalian cells. We took as our example short RNA, a 67 nucleotide short hairpin RNA called FF1. Now, in order to measure concentration of these short RNAs in vivo, we used an indirect readout. Here we have constitutive promotion of EYFP 4XFF1, which is a yellow fluorescent protein with 4 FF1 binding domains on its mRNA. After being processed, FF1 can bind these binding domains, and through RNA interference, knock down production of the EYFP protein. We quantified this using flow cytometry. Here are sample traces from one of our experiments. On the x-axis, we have relative intensity in the FITC channel, which measures yellow fluorescence. Here we can see that when there is no FF1 present, there is a significant population shift to the right, indicating yellow fluorescence. As we increase the amount of FF1 produced, we see that the population shifts back towards the left, indicating protein knockdown. We further quantified this by repeating the experiment in triplicate. Here on the y-axis, we have mean fluorescence over 10,000 cells per experiment, and on the x-axis, we have amount of FF1 DNA put into the cell. We can see that there is a marked decrease in yellow fluorescence, indicating that our FF1 RNA is being produced. Now that we have a method of production of our circuits, how do we interface with the cell? We chose to sense mRNAs because mRNA levels are correlated with cell phenotype. In addition, the transcriptome interfaces nicely with our RNA gates. We designate a section of our input mRNA to serve as a toehold and hybridization domain in our strand displacement motif. And we can design a specific sensor or gate output complex for this sequence. There are a couple design rules that we have to follow, the first of which is orthogonality. We want our sequence to be completely orthogonal to other mRNAs in the cell, such that our sensor is highly specific. In addition, we adhere as closely as possible to a 3-letter code to minimize secondary structure in our signal. The toehold region on the input mRNA binds to the gate strand and displaces the output, which then serves as a downstream input. We can further amplify our signal by adding a fuel molecule, which displaces the mRNA from the gate strand and allows this mRNA to further interact with more sensor molecules. There is a third design rule that we kept in mind, which is that of accessibility, which I'll address by showing you an example mRNA. We conducted experiments with eBFP2, which is shown here in a software rendering. As you can see, mRNAs have significant secondary structure, including stems, loops, and bulges. Let's take a closer look at one region that we chose to sense. Here in blue, we have the toehold region, and in green, the hybridization domain. We rapidly prototyped our mRNA sensors using DNA oligos and coupled the sensing reaction to a downstream fluorescent readout reaction. This means that if we do sense our mRNA, we should see an increase in fluorescence. We chose 4 regions on eBFP2 that we designed sensor molecules for and conducted our experiments on a plate reader. In black, we have baseline fluorescence, and in red, we can see a 2- to 3-fold increase for each of our 4 sensing domains. This means that we are sensing our eBFP2 mRNA.
Now that we know that it's actually feasible to do useful things with strand displacement inside mammalian cells, we want to know whether strand displacement can actually happen inside of a mammalian context versus in previous in vitro studies. We again look at the buffer and simplify it a bit. We turn it into a fluorescent readout where we again have a quencher-fluorophore pair such that when the quencher is close, the fluorophore is quenched. And we add a small green dye so we can actually track this complex all the time. We expect this complex to be green, but not red because of the quencher. However, when an input strand binds, it can displace the strand with the quencher, now allowing red fluorescence. Thus, we expect this strand to be high in green and red fluorescence levels. It turns out no one's used these modified RNA strands for strand displacement before. So we actually have to verify that this reporting mechanism works with RNA. We did this in solution using a plate reader and measuring fluorescence over time. Here, we see our negative control. Here, the incorrect input strand doesn't have a matching domain to the reporter, so it cannot displace the quenching strand. We expect to see no fluorescence increase over time. And that's exactly what we see. Red fluorescence is low all throughout the experiment. We look at our positive control of just the strand with the red fluorophore and see that fluorescence is high throughout the experiment. Finally, we add our input strand, and we see that fluorescence increases over time as strand displacement occurs. This is because the input strand comes in, displaces the quenching strand, and now allowing separation between the quencher and the fluorophore, resulting in increase in fluorescence. The next step would be to deliver these reported complexes to mammalian cells. So we did just that. We visualized our cells and visualized the RNA. Here, we look at HEC293 cells that express a yellow fluorescent protein. These are mammalian cells. And we delivered to it an RNA strand with a red fluorophore. In this video, you'll see the yellow cell that is then approached by a red vesicle containing our RNA. The vesicle attaches, fuses with the membrane, and then delivers its payload, indicated here by this increase in whole cell red fluorescence. Over the summer, we also optimized the delivery of double-stranded RNA, as well as a combination of single-stranded and double-stranded RNA. now we are able to actually measure strand displacement happening inside a mammalian cell. We did this by measuring the fluorescence using flow cytometry, where we plot both green fluorescence, shown here in the Fitchy channel, and red fluorescence here in the Texas red channel. We started with the population of cells that did not receive any RNA, and we see that there is low autofluorescence. We then look at our reporter complex and see that this complex is fluorescent green, indicated by the population shift to higher green values. Our positive control looks like this. This is just a double-stranded complex with a red fluorophore. We see that this population of cells has shifted towards higher red levels. Finally, when we add an input strand and a reporter, we see that there is an increase in red fluorescence levels due to strand displacement happening. We see that these cells are both red and green. However, we look at this and we compare our reaction to our double-stranded ROX complex. This is the positive control. And we see that we can actually optimize this a little bit further. So we think about the parts of the reporter that we can actually optimize. We can think about the sequence of the reporter and make it orthogonal to our host cells, such that we don't have any false positives. We can think about the toehold length, which will determine the strand displacement kinetics, and the domain length, which will determine the stability of this reporter complex. I also mentioned the fluorophores that we can use to actually have a readout of strand displacement, as well as to track our strands. We can also make certain modifications to protect our strands. Now, in our second iteration, we chose to use a slightly longer toehold, going from 5 nucleotides to 8 nucleotides, indicating faster kinetics, and a longer domain, including now orthogonal sequences to the HEC293 cells. We also used an improved delivery method. Here we see our negative population again. Now we see a reported population that is just fluorescent in the green channel. Our positive control looks like this. And most importantly, when we add our inputs, we see that this population has now significantly shifted compared to the no input population. We can even look at this slightly differently and plot the two populations on this graph where we show red fluorescence and the number of cells at this particular fluorescence level. We see that there are two distinct populations, the no input population, shown here in green, and the input population, shown here in blue. We can quantify this by putting a number on the fold increase in fluorescence due to strand displacement happening. And we see that there's a 6-fold increase in the mean fluorescence levels. So after a few rounds of optimization, we achieved our goal of showing that strand displacement can occur inside mammalian cells. We did a lot over the summer. We showed that we have a functionally complete set of logic operations, that we can actually produce RNA circuits inside cells, that we can interface with the cell, and finally, that strand displacement, the key reaction behind it all, does actually occur in mammalian cells.
So now that we've shown that strand displacement is a viable information processing technology, let's take a closer look at a potential circuit that could be enabled by our results. So let's consider these 6 target inputs that are indicative of a cancerous cell state, specifically a pancreatic cancer cell. And these 6 inputs are messenger RNAs, where 3 are expressed high inside of the cell and 3 are expressed low. And we can interface these mRNAs directly with our mRNA sensor that we designed, developed, and tested, and with our strand displacement inverter. We can then process these signals using cascades of strand displacement reactions using AND and OR gates, and then transduce this nucleic acid signal into a protein output, for example, by translating a killer protein that would kill only the cancerous cells with this specific messenger RNA expression profile, or a fluorescent protein, which could be immediately used by doctors as a diagnostic tool. However, we believe that this is just one potential application of strand displacement. This technology can really transcend multiple application spaces. And all the results that we've shown to you today can be replicated in your lab because we've submitted all of our parts to the Registry of Standard Biological Parts, and you can check them out online. As well as we believe, to ensure the future of synthetic biology, we really need to educate people of all levels. So we started off young by teaching a middle school class at summer HSSP. We bolstered our curriculum and developed a high school class, which we are teaching in 2 weeks, about SynBio, and we hope to inspire this high school to form their own iGEM team, which we will mentor one day. As well as we've developed an undergrad class that we teach in January at MIT, available for all undergrads in the Boston area, about how to engineer luminescent bacteria. As well as we have a very strong collaboration with Wellesley's iGEM team, and we've been a test case for their software all summer, testing their MoClo planning design. At this time, we would like to thank our faculty advisor, Professor Ron Weiss, as well as all of our mentors, our very generous sponsors, and now I'd like to invite our whole team up to the stage, and we will take any questions that you might have. Thank you.
I'm going to pose the first question. So you said that you designed a NOT gate in vitro and that you made actually 2 designs and you simulated the output and only one worked. And I was just wondering what was the difference in the design? So what made the one work and the other one not?
So there are multiple components to our NOT gate. There's the let's just actually look up this slide. So there's a catalytic cycle that allows us to actually do the signal processing as well as this cooperative hybridization gate that Julia might explain. So
the main difference in the 2 design is due to one molecule, the molecule that is responsible for the behavior of the NOT gate. That is molecule A, that is responsible for the behavior of the NOT gate when the input signal is high. So in the previous design, we used, instead of this molecule that has 2 strands, another molecule that has 3 strands. And unfortunately, with that kind of molecule that was, first of all, more complex of this, we were not able to have a low output when the input signal was high with simulation. So we discarded the design, we arrived here, and we saw that simulating this design, we were able to have a low output when the input was high. So for this reason, we went ahead with this design, and we went ahead with the experiment with this design.
So there are a few papers for in vivo strand displacement. So would you tell me the strong point of your system?
So our method of strand displacement actually includes the whole toehold part. And actually, this specific mechanism of toehold mediated strand displacement has been shown to be very successful. We showed you this a square root calculator circuit before that used 12 gates and many components. And we believe that this specific implementation of strand displacement is very powerful, and that's why we chose this implementation.
Thank you. And another short comment. And you used 3 letters to avoid secondary structure of RNA. So can you combine your system with unnatural base?
With unnatural bases?
Yes.
This is something we haven't explored yet, but it's definitely something to think about. We know that we can actually do RNA to DNA interactions, so it might be possible, depending on how many foreign bases there are. I believe.
Thank you.
Is there a potential concern for immune response to double-stranded RNA like RIG-I, TLR3, and others?
Yes, that's true. There are, but we envision our system to be actually continuously produced as well as decayed. So we have a system to produce short RNAs, and we also show that there are secondary structure elements, not just chemical base pair modifications that actually give you resistance to these pathways. We've seen papers that show that adding a small bulge into a double-stranded complex can actually confer resistance to some of these pathways. So we envision that by using a complementary approach of both having transcription produce these circuits continuously, as well as the secondary structure elements, we can actually protect our RNA from being degraded, while keeping degradation an important aspect of our circuit.
So how do you envision the therapeutic use of this system? You would introduce it with a virus or
well, current technologies have been using viruses very successfully, and we envision that since we have the very small footprint for our circuit, we can actually put a lot of sophisticated circuits into a viral vector and then deliver it. Yes, while we focus on the information processing aspect right now, we do envision that viral therapeutics could be a solution. But there are also other ways like nanoparticles and so on that are all being researched right now. So thank you.
Other questions from the audience?
So you have tried your key reaction in the cytosol of your mammalian cells, but probably natural RNA processing happens inside the nucleus. So have you tried anything with naturally transcribed RNA in the nucleus?
So we currently do have designs for this, but we have not had the time to run the experiments. We did look at the U6 promoter to produce these short RNAs, and we do expect that these RNAs will now be in the nucleus for our processing.
Just one quick question on the experimental point of view. Did you do the modification of the mRNA yourself, adding the quencher or by yourself, or did you order the RNAs with the quenchers and the fluorescent tag?
The RNA with the quencher and the fluorophore were ordered by IDT, and we have these sequences up. You can just enter the order to IDT, and you can order them
Right,
yourself.
so you just order the oligos and transfect them, right?
Yeah.
Right. So it's not really in vivo thing.
I mean, in vivo,
sense.
the in vivo step is the actual process of strand displacement happening inside a vivo system. This has not been shown before. We've been looking at control systems where we have controlled magnesium concentrations, no enzymes, and so on. So the in vivo aspect is still there.
Yeah.
Are there more questions? And thanks again.
Thank you.
MIT.